All right, so for the last part of chapter 7, that is going to be Calvin cycle and then some modifications plants have. We're going to talk about those as well. But let's start with the Calvin cycle. And so again, the Calvin cycle, we have the NADP plus and ADP needed for the light dependent reaction comes from the Calvin cycle. And again, this is a big cycle between the two. And so we're going to take the NAD. So here's our sun, right? We've split our water. We just talked about that. We have that NADPH. So we have the NADP plus coming in, picking up electrons, and then it's going to take and dump them off in the Calvin cycle. The same with ADP. ADP we're going to use our energy here. We're going to take our empty ADP, fill it back up. And then again, this is an anabolic reaction or endergonic, meaning it needs energy. And so we're going to get that energy from the light reaction. There are 3 stages in the Calvin cycle. There is carbon fixation, there is reduction, and regeneration. So we're going to talk about those 3 cycles. So with carbon fixation, when we talk about things being fixed, it's like, fixed versus being broken. So when we talk about CO2 is inorganic. We can't use it. So we need to fix it. And what I mean by fix it, we need to turn it into something that is organic. And so what we're going to do is we're going to take the CO2, we're going to hook it up to a molecule called RUBP. We are going to use an enzyme. That enzyme is Rubisco. And so Rubisco is going to take RUBP and that CO2, and it's going to add it. You can see how it added it on to RUBP. And once it does that, it cuts it in half. It splits it in half. And we get 6 PGAs. I always think of Professional Gulf Association. That's our first product here in our circle. PGA. Once we have our PGA, we are then going to reduce it. Okay, so we're going to take and put, because think about it, with the CO2, we have sugar, which is C6H12O6. So for every carbon, we have a molecule of water. Well, I just added carbon and I just added oxygen, but I did not add hydrogen. So where do I get the hydrogen? I get the hydrogen from NADPH. So I'm going to take that NADPH and I'm going to put that hydrogen onto my PGA, and then I'm going to make what's called G3P. Remember, I got 6 of those. Okay. We're also, because this reaction is endergonic, we need power, we need energy, and we're going to use that ATP that we made in the light reaction. And now we have 6 molecules of G3P. 1 molecule is going to exit the cycle, and we're going to use that as a 3-carbon sugar. We're going to use that to make all kinds of biological macromolecules inside the plant. Then we're going to take the remaining 5 G3Ps and we're going to rearrange it. And that also needs more ATP. So we need more ATP from the light reaction to regenerate our RUBP. All right, so here is that whole cycle. So we start with RUBP. We're going to fix our carbon dioxide with the help of our enzyme Rubisco. We're going to add it because these are 5. This is a 5 sugar, right? 1, 2, 3, 4, 5. We're going to make it into a 6-carbon sugar. We're going to split it in half. And now we have 1, 2, 3, 4, 5, 6 of those molecules called PGA. We're going to use some energy, and we need some hydrogens because we added carbon and oxygen. We're going to add the hydrogens. And now we have 6 of the same molecules. We're going to exit 1 to make our biological macromolecules. And then we're going to use the other 5 and some more energy from the light reaction to regenerate RUBP. All right, so our inputs are NADPH. What do we get out? NADP plus. And again, remember, then that's going to go to the light reaction. I'm just going to go LR light reaction. Then we have our ATP. We have our ADP plus our phosphate. And guess what? That's going to go back to the light reaction. And then we put in our CO2, right? Plants take in CO2, and we're going to make our G3P, which is going to go to make other biological macromolecules. We're going to make our sugars. We're going to make our lipids. We're going to make our amino acids. So here's kind of that overview of things. So we see our light reaction, we're splitting our water, we're energizing going into ETC, we're building up our concentration gradient so we can make our ATP. The ATP will be used in 2 locations in our Calvin cycle. And then we're going to take our electron, give it more energy, and we're going to make our NADPH. We're going to deliver that hydrogen onto our PGA. And then once it's empty, that's going to go back to the light reaction. So it's a big old cycle here between the light reaction and the Calvin cycle. And basically, when we look at it, all we need to do is put in some water, some sunlight, and our carbon dioxide. Right? Carbon dioxide, water, light, and we end up with sugar and our waste product, oxygen. So we have a problem because Rubisco has been around for a very, very long time, and it was around before there was free oxygen in the atmosphere. Now that there's free oxygen in the atmosphere, Rubisco can also fix oxygen, meaning it can combine with either oxygen or carbon dioxide. So oxygen would be considered like a competitive inhibitor. So when we have situations where we have hot and dry environments, we have what's called the stomata. The stomata is the opening. I don't remember if they show you a picture. They do not. Let me see if I can get this to draw. The stomata are, and you guys should hopefully remember from your lab, are these pores made by 2 cells called guard cells. And the pore lets in oxygen and lets out. Ooh, I just totally did this backwards. Let me just do this. I was too busy to concentrate on what colors I wanted to use. Let's just do this. Let's in our CO2, right? Let's add our oxygen. But another problem is when it's very hot and dry, it can also let out water vapor, and we don't want to lose water vapor. We lose water vapor. Remember, we need water for the light reaction. And so what can happen is when we have those situations, the stomata will literally, when it's losing that water because turgor pressure goes down, they'll sag and they'll close the pore. And so if the pore closes, what happens is the inside of that leaf will increase in oxygen, right? As it's still photosynthesizing, but it will decrease in CO2 because the CO2 can't get in, oxygen can't get out. And so now Rubisco has a higher chance of binding with oxygen. When it binds with oxygen, it's counterproductive. So what happens here, we call that photorespiration. So when we bind to oxygen, what happens is we still use the NADPH, and we still use the ATP from the light reaction, but we do not produce any, notice, no sugar, no G3P. So we're using all those energy molecules from the light reaction, but we're not producing any food. So it's counterproductive. We call this photorespiration because, right, respiration, you take in oxygen. So it's taking in oxygen, but it's using the products from the light reaction to make no food. So it is a negative process for plants. So here's another picture. So photorespiration, again, we're gonna take in oxygen and give off CO2. We're not making any sugar. You know, if RUBP binds with Rubisco and CO2, we end up making the food. So that's photosynthesis versus photorespiration. We don't want to do photorespiration. So plants have had to modify some of their methods of doing the Calvin cycle to prevent this loss in product. And so there are a couple different ways through evolution that we have adapted some kind of additions or modifications to avoid photorespiration. For one, in some plants, they have compartmentalized some of the locations in which they are going to do the, we call the regular Calvin cycle the C3. That's another name for the Calvin cycle versus the C4. C4 is another mechanism that we have carried out. And so what we're going to do is when the guard cells are open, we are going to undergo a C4 process that basically stores carbon dioxide. And so as we store that carbon dioxide, when the stomata close and we have low amounts of CO2, it can re-release that carbon dioxide to be used. So here are some of the modifications. So here's C3. C3, we're just going to go through our normal process. We're going to take in our carbon dioxide, go through Calvin cycle. C4, notice they've compartmentalized now in their mesophyll versus their bundle sheath cells. They're going to do a Calvin cycle in their bundle sheath cells. They're going to do C4 in the mesophyll, and they're going to take in the CO2 and store it as a 4-carbon molecule. So what happens is when C4 plants are going to close their stomata, they're going to purposely close their stomata in the hottest part of the day to prevent water loss. If we don't have water, we can't do photosynthesis at all. So it's going to close those stomata so it doesn't lose the water. And then remember, it's still getting water from the root system. And so then, because we're more exiting the water on the stomata than taking it in. We're taking the water in through the roots. And so once it closes its stomata, it has this stored CO2 it can release to help reduce the probability of Rubisco binding to oxygen versus CO2. Now, CAM plants are a little different. CAM plants also have that compartmentalization, but they only open their stomata at night. So these are plants that live in very, very hot and dry temperatures, like deserts. These are we call this the crustaceation, I can't even pronounce it, acidic metabolism. So we always call it CAM. It's just CAM plants. So they're in very hot and dry environments. So this would be our cacti. And so they only open their stomata at night. So they open their stomata at night, they let in the CO2, they store it as that C4 molecule. And then during the day, because they need the ATP and NADPH driven by the light reaction, they will then undergo photosynthesis and build sugar during the day. So they only have a very limited amount of carbon dioxide, which means they're very limited to the amount of food they can make, which is reflected in how slow they grow. All right. So again, this is a modification that allows plants in the desert, but they are slow growers. So this is the end of the chapter. So we talked about photoautotrophs, photosynthesis. We talked about the 2 stages. We talked about the light dependent, the light reaction, the Calvin cycle. We talked a little bit about wavelengths of light. We talked about the photosystems. We talked about carbon fixation, and we talked about evolutionary adaptations to avoid photorespiration. So I hope that was helpful for you. Again, there are more videos in Canvas for you to watch, and you can always ask me questions in lab. All right, I will see you in the next video.